Okay, I'm here now recording chapter 5 for us, going over the genetical theory of natural selection chapter that we were unable to cover by Zoom on Thursday. I apologize for taking a while to get this up. And I will have chapter 6 up as well within the next day or so, assuming I have power, given that we've got Snowmageddon hitting Oxford. Okay, so the whole purpose of this chapter is to relate your understanding of evolutionary change to this point to the genetic basis of the variation that we now know exists within all populations. That variation arises due to mutation, but it's critical that we focus early on here on the link between these genetic variants that exist, these different alleles for a particular locus within a population, their phenotypes that they cause, the different phenotypes that appear in, say, these moths on the left side of your screen, and then how selection acting on those phenotypes of those individuals in the population can then, because of the heritability of the alleles that cause those different phenotypic traits, the population can change over time if those different phenotypes have different fitness. And by fitness, I mean survival and reproductive success rates. Okay. So this example is one drawn from post-industrial England. And when the Industrial Revolution hit England, particularly in very highly populated areas like London, the soot, the smoke output was pretty significant. And what it did was coat the trees in the neighboring areas that are normally covered with this living material we call lichen, as you see on the right image. And it creates this kind of mossy look on the trunk of trees. And the peppered moths that live in that area were predominantly white with black flecks on them. Okay, you can see in these images there are two different forms of peppered moths, one that's solid black and another form that is kind of this peppered coloration. Before the Industrial Revolution, these peppered colored moths represented the vast majority of the individuals in the population. More than 90% of the individuals had this peppered coloration. And that's because they blended in better on the trunks of trees that they would rest on. These black moths, as you can see, they possess a mutation that causes the overexpression of melanin, this dark pigment that creates this entirely black coloration. Well, before the Industrial Revolution, those black moths that existed in the population occurred at a low frequency, and that their frequency stayed low because they had lower survival rates, right? Birds more easily see them, consume them, so they're eaten at a higher rate. They have offspring at a lower rate. They don't stick around in the population. But with the Industrial Revolution and the soot doing two things, killing the lichen and covering the trees in soot, all of a sudden, as you can see in the left image, the black moths blend in quite a bit better on these non-lichen covered or soot covered trees. The peppered moths fared poorly. Okay, they now stick out like a sore thumb. They are now eaten at a higher rate. They have lower survival success. They have on average fewer offspring than their black counterparts within this population. And so the frequency of black moths in the population increased dramatically to the point where in the early 60s, nearly 100% of the population, or three populations that are depicted by this graph on the right, a red, blue, and green population. These three different locations, the black moths represented 90 to 100% of the individuals in these populations. But as time went on, between the 60s and the recent times in the 2000s, the frequency of this black form has decreased dramatically from nearly 100% down to less than 10%. Okay. And this is because since the Industrial Revolution, the output of soot from engines has that has been refined and reduced. And so lichen is growing on these trees again. And once again, the peppered form of the moths has a higher survival rate, and they are thus increasing in frequency in the population. And if that's happening, necessarily the black phenotype is decreasing in frequency. So this really encapsulates a study of natural selection acting on a population that has genetic variation. That genetic variation is heritable and causes differences in phenotypes, okay? Different phenotypic variation. And selection is acting on the phenotypes of these moths, and therefore the population is changing over time. Another example your text presents is an interesting one, these being German cockroaches, the small cockroaches that you might see in a dorm room or the house of a bunch of young undergraduate males that are averse to perhaps cleaning the kitchen all that often. Or females, no judgment there. We're all messy sometimes. The results of natural selection in this example are pretty remarkable because what we've seen is in recent decades, these roaches have evolved to avoid consuming the poison that are in these commercially available roach traps that you can buy at Walmart or Home Depot. And so the graphs here are illustrating neuron responses of two different neurons in these cockroaches to things that they taste that are sweet or bitter, right? So cockroaches have a particular neuron, illustrated on the right side of these graphs, that fires when they taste something sweet. They have a separate neuron on the left, illustrated in these graphs, that fires when they taste something bitter. They don't eat bitter things, okay? Now, fructose and glucose are both sweet, and when cockroaches that are wild type, that don't have this mutation we're going to talk about, if you go catch a German cockroach out in the wild, they're most likely to have these responses that we see to these three different items, fructose, caffeine, and glucose. If you give a wild type roach something with fructose in it, only its sweet neuron will fire. The same will happen if you give it glucose. But if you give this same roach caffeine, the bitter neuron will fire and it will go away. It will not attempt to consume whatever that is. Now, the mutation that has arisen within populations of these roaches that live in human houses is one that has helped them avoid death by roach trap. Okay, they have the same response to the sweetness of fructose. They have the same averse response to the bitterness of caffeine. But interestingly, they have a mutation that causes their bitter neuron receptor to fire when they are exposed to glucose. And so it might not surprise you to learn that glucose is actually the material that is put into roach traps. And so these roaches in your house, if you have roaches in your house that are not dying because you put out these roach traps, they may possess this mutation that causes them to avoid consuming whatever's in those traps because it has glucose in it. It causes the bitter neuron, in addition to the sweet neuron, to fire. But that's enough to keep them from tasting that material, okay? And so think about how this population has evolved over time. What has happened is that this mutation arose, and those individuals that possess the mutation had a higher survival rate because they never eat the poison. They are going to have more offspring. They are going to live longer than individuals that don't have that mutation. Okay. So these same principles apply to instances of artificial selection, where humans make the decisions about which individuals survive and have high reproductive success, right? So think about how Great Danes were bred into being so large, or Chihuahuas so small. When a breeder of teacup Chihuahuas has puppies, and they're considering keeping one of those puppies for the next generation to breed, they might sell a bunch of them, but which one are they going to keep? Are they going to keep the biggest teacup Chihuahua? Well, that's not the point of teacup Chihuahuas, right? They're going to keep the smallest one so that they can have the teacuppiest, tiniest, most ridiculously small Chihuahuas. Okay. Same is true of Great Danes. Those breeding Great Danes, they are selecting the largest individuals as breeders for their next generation of offspring. The same is what has led to the dramatic changes you can see here in the size of chickens that are used and are bred for meat production. Now, I've purchased these chickens quite frequently and raised them for meat, and it's a whole different ballgame compared to a normal chicken, say one that you get to lay eggs. Okay. Now, this breed of chicken has evolved very rapidly. Since the 50s, you can see that back in the 50s, when they're born, they averaged 34 grams. And at 56 days, they were just under a kilo, 905 grams. And that's not all that different from a normal egg-laying chicken, right? They grow pretty slow. If you went to harvest that chicken at 2 months, it's not much of a meal, okay? But through artificial selection over the past 50 years, these chickens now average at 56 days over 4 kilos, about 8 pounds. That is an enormous chicken. So the next time you're driving down the highway and you see one of those semis pulling all these crates of white chickens that are just sitting there and not doing a whole lot, that's what these are. They're called Cornish cross. It's a breed of chicken that is bred specifically to grow incredibly fast, and they don't get up and do a whole lot. They convert food to meat as quickly and efficiently as possible. Now, a lot of times, as I've interacted with people who raise and breed chickens, because as I said, I own chickens and I've raised these chickens in the past for meat, you'll hear kind of purists or super, let's say granola type people claim that these are genetically engineered or GMO, right? There's a very big difference between genetically engineering something and artificially selecting for something, okay? So something that's genetically engineered is the corn or the cotton that we grow here in Mississippi. So when you drive by a corn or a cotton field and you look at how beautiful that field looks and there are no weeds out in that whole damn field, okay? That's because they have genetically engineered, they've put a gene, some DNA from a bacterium into that plant, that corn or that cotton. And that gene allows you to spray weed killer, plant killer, herbicide on that entire pasture, that entire field of corn or cotton. And those plants don't die because of that bacterial gene that's been genetically engineered in there. That is actual manipulation of the genome of those plants. What we're talking about here is something very different. Nobody made these chickens in a lab by manipulating their DNA and putting growth hormones in them, okay? What has been done to these chickens is that they've been selectively bred, just like a Chihuahua or a Great Dane, over many generations to grow fast and produce, just put on as much weight as fast as possible, okay? So there's a big difference between artificial selection and GMO, all right, which we'll get into in a later class. All right, so what happens to a population that has variation in size, okay, if there is or is not selection? So we've been talking about populations that are variable, right? And that variation is heritable. So in both of these, we are seeing a population where the average size of parents on the x-axis can be determined, and parents can be really small, far to the left dot, or very large, far to the right dot, okay? And the reason there is a linear relationship, bottom left to top right, for all these dots, is that wherever on the x-axis, so for however far right you go on the x-axis, you go that high on the y-axis. Whatever the average of the parents is what the average of the offspring is going to be. Okay, that's why all those dots are on that diagonal. Now, if there's no selection, meaning the population's variable, that's great, but there's no selection. Everybody has the same probability of reproductive success. Everybody has the same survival rate. Everybody survives. Everybody has offspring. Then there's no change, right? The offspring are going to be offspring from all of these dots. Every one of these parents, these dots representing a parental pair, will have offspring, and those offspring will have the same average as their parents. Now on the right, what is illustrated is the same population, but here we've got selection where only the largest individuals survive. Okay, so the population has the exact same variation in it, but when selection removes every individual parent that is smaller than that dashed line, okay, every parent to the left of that dashed line dies. Who survives to reproduce? Only the solid red dots that are to the right of that line. Okay. And so if only those really large parents reproduce and their offspring are going to inherit those alleles for large size from them, there's a one-to-one relationship. There's a very clear relationship between parental size and offspring size. What that means is the average of the offspring is going to be pretty high, the average size. And it's going to be higher than that original parental average, where this P is down on the x-axis. That was the original average of the parental population, okay? But because of differential survival, the average of the parents that actually bred and had offspring is shifted far to the right, okay? So when selection acts on these parents, the average of the offspring will change. And this is that shift, that movement of the population over time due to selection. So when we think of the fitness of individuals, it's important to recognize that fitness is in equal parts survival and reproductive success, right? Now, it's tempting to think about those things in the context of survival from the time you're born, right? And then reproductive success during mating season, okay? But there's a lot more to it, so that fitness, there are components of fitness that come into play throughout the life cycle, even at the stage of a zygote, right? So on the top left there, fertilization success, there are alleles in, say, sperm that might make them less likely to fertilize an egg than another sperm that has other alleles, okay? So this is an example of fitness acting on gametes, the alleles within gametes. Okay, so survival of that gamete is dependent on it joining with an egg. And if that doesn't happen because of lower fitness of that particular gamete, because of alleles that it possesses, right, that's a component of fitness. The viability of zygotes, right, there's a significant period of the life cycle where individuals are simply zygotes, right? They have not yet been born. They're not yet growing outside of their parent, whether it's an egg or in the womb. And so that period of viability affects the fitness of individuals and can be determined by the genotype of the zygote. Okay. Now, once the individuals are adults, mating success, we've talked a good bit about that, determines whether or not those individuals become parents. Once they do reproduce, fecundity is a big player in the calculation of fitness. And fecundity refers specifically to the reproductive output. How many offspring do individuals have, right? So they're different individuals with different genotypes may have very different fecundity, right? So two females might mate exactly the same number of times, but one female has 10% more offspring on average, okay? So we have mechanisms for calculating or quantifying measures of fitness. And we're going to talk about two of them, and they are related to one another. So the first one is the absolute fitness of a particular genotype. This is a pretty simple calculation. It's simply the product of probability of survival of the individual genotype or the average probability that an individual with that genotype would survive, times the average number of offspring for that genotype. Okay? So the homozygous dominant genotype illustrated here has a 70% chance of survival. So every individual of 10 individuals born with that homozygous dominant genotype, 70% are expected to survive, 7 out of 10. Now, those that survive will have on average 324 offspring. Okay? Now, the homozygous recessive genotype, individuals with that genotype have a 90% chance of survival, and they on average have 139 offspring. The absolute fitness is a number that is calculated by multiplying these two numbers, and you can see I've calculated those here, and you can compare directly the absolute fitness of the homozygous dominant and homozygous recessive genotype. But you need to realize that, this is going to be different for every organism, right? For a human, those numbers on the right are not going to be as large. They're going to be 1, 2, 3, or 4, right? So what's the difference in fitness between 226.8 and 125.1? How much better is that? And how much better is 220 than 125.1? Okay. So these absolute fitness numbers are helpful, but they're difficult to compare one another. All right. It's difficult to get a metric for how much better one is than the other. So we need to consider them into the context of a way in which we can compare them equally. And we do that by calculating the relative fitness. And the relative fitness is a measure that takes that measure of absolute fitness. Those numbers on the left here, 226.8, 220, and 125.1 are the same as the previous slide. Those are the absolute fitness. We take the absolute fitness of every genotype, and we divide it by one number. We divide them all by the same number. And that number is the absolute fitness that is the highest of all. Okay? In this case, that's 226.8. So when we calculate the relative fitness of the homozygous dominant genotype, we divide 226.8 by 226.8. Its relative fitness is 1. Okay? So the genotype with the highest absolute fitness is always going to have a relative fitness of 1.0. Okay? It's the point of reference. Now, the heterozygous genotype, you divide it by 226.8 and you get 0.97. You divide the homozygous recessive genotype's absolute fitness by 226.8 and you get 0.55. These three numbers are the relative fitness, but what they tell you is that the heterozygote is 97% as fit as the homozygous dominant genotype. The homozygous recessive genotype is 55% as fit as the homozygous dominant genotype. Okay? So if you're given these measures of relative fitness, what is the difference between 0.55 and 1? Okay? Well, that would be 0.45. What that means is that selection is acting against that homozygous recessive genotype. It's taking a 45% hit on its fitness because of selection. Okay? The heterozygous genotype is taking a 3% hit or 0.03 hit. Selection is acting against it that much. Okay? So let's think about how a population responds when selection acts against a particular allele. Okay, in this case, we have two alleles, A1 and A2. They occur at equal frequency in the population. Okay, look at the far left, all right? We have 8, 10, 12, 14, 16 copies of A1 and 16 copies of A2, right? They're perfectly equal in their frequencies, okay? Now, there are more heterozygotes than there are of either homozygote, but that's not the point here. All right. So what we're going to see is that as these juveniles grow up, because selection is acting against A1, we're going to see half of those A1 homozygotes die and a quarter of those heterozygotes die. Okay. This has changed the frequency of the genotypes. Compare the bar graphs on the left and in the middle. Okay. Now, when these surviving adults are the only ones that produce offspring, what you'll realize is that in the surviving adults, there are more A2 alleles proportionately than there were when this population consisted of all the juveniles. And because A2 is now higher in frequency, it is more likely to be passed to offspring. More A2 alleles will go to the next generation than A1s because there are more A2s in this population of surviving adults. And so the next generation of offspring has a higher frequency of A2 homozygotes, and there is a higher frequency of the A2 allele in this population. Now, the rate at which this A2 allele frequency changes is highly dependent on how strongly selection is acting on this gene, okay? So in this case, selection is acting for A2 or against A1, okay? And the strength of selection can vary, okay? The strength of selection could be 0.01, 0.02, 0.05. And I want you to think back to our relative fitness calculation. This is a strength of fitness. This S number is basically what is lost in that relative fitness calculation. And so if S is 0.01, what that means is that selection is causing A1 to have 1% lower fitness, or 2% or 5% lower fitness, okay? So the reduction in fitness of the allele A1 that is caused by selection can be 1%, 2%, or 5% in this example. And what you're seeing is how quickly will the frequency of A2 increase at those different levels of selection. So if selection confers a 5% advantage to A2, then within 100 generations or so, well before 200 generations, A2 is going to be 100% of the population, right? The population will become fixed for A2, and that means only A2 is present. If it's 5% better, then that's a lot better, and A1's going to be gone relatively soon in fewer than 200 generations, okay? Now, if A2 confers only a 2% advantage, you can see it takes over 400 generations. If it confers only a 1% advantage, it's really slow, hundreds and hundreds, perhaps thousands of generations before the A1 allele is gone and A2 reaches a frequency of 100%. Now, we've got some really cool examples of this type of selection happening in the natural world, and this is actually within humans, okay? So, as you're probably aware, humans are unusual in that they continue consuming milk well into adulthood as mammals. That's pretty uncommon. What's interesting is that we can trace this back to pastoral societies in which cattle were domesticated in Northern Europe in the area demarcated by the dashed line. So in that area, okay, because people were domesticating cattle and raising them for milk and dairy products, selection acted quite strongly, 10% strength of selection, on favoring the persistence of alleles that allow humans to continue to digest dairy products or break down lactase. Okay. So, and in that area where these societies existed, you can see that that is where the frequency of these alleles is the highest. Okay. So selection acted on the human population. Hey, there's this really easily accessible source of protein, mutations that allow you to access that throughout your life have a selective advantage, are selected for, and the frequency of those alleles increase dramatically over time. The same is true of alleles that confer lighter skin coloration, okay? What's interesting is these alleles arose, or alleles that contribute to this lighter skin coloration, arose as humans moved north, where sunlight was less and less available, right? It was, you have shorter days, longer nights, less sunlight available to produce vitamin D, okay? As you are probably aware, we produce vitamin D when we are exposed to sunlight, okay? And if we don't get sunlight, we don't make our vitamin D, okay? So as populations moved north into Europe and Eurasia, they had to evolve lighter skin coloration so that they could absorb more of the sunlight and produce more vitamin D, okay? Now you'll notice that the blue pie charts indicating a very low frequency of the light skin color allele in Central Africa, where skin coloration is darker. That's what you would expect to see. What's a bit surprising is that in Eastern Asia, that light skin color allele is also quite rare, okay? But skin color in that region is quite light. And they are also subjected to the same lack of sunlight. And so it probably won't surprise you to realize or to learn that these regions in which populations do need and actually have a mutation that allows them to access more sunlight because their skin is lighter, it's actually a completely different mutation that allows those individuals to have lighter skin so that they can absorb and process more sunlight, produce more vitamin D, okay? So again, this is an instance of a mutation that arose in a variable population. As you can see, that allele is present in Central and Southern Africa, but it is not beneficial there. When these populations moved north, it became beneficial, increased in frequency. So when something like this arises in a population, or the environment changes and creates an environment that is advantageous for a particular variant, does that mean that that mutation is going to immediately rush to fixation? Well, let's just imagine how big the human population is, right? You are one of 330 million people just in the United States, right? One of many billions of people on the planet. Let's say that you, right now, you are a carrier for an incredibly advantageous mutation, okay, that gives complete resistance to coronavirus infection, right? That's awesome. Everybody should have that. We all want that. And you're the only one that has it in the whole world. Now, you probably only have one copy of it, right? Let's say your mother gave it to you. There was a mutation as she was creating the egg that became you. Father didn't have it, so you're heterozygous. How many offspring are you going to have? 1, 2, less than 25, I would imagine. Not a whole lot in the grand scheme of billions of humans on this planet, okay? So let's just say you have 2 offspring. What proportion of them are going to get that allele? If you think about how gametes are made, right? Your mom gave you this beneficial allele, dad gave you the normal thing that all the rest of us with coronavirus have. So you've got 2 different things. You have 2 offspring. On average, you're going to give one of those offspring the good new mutation, the other something else, okay? So that's it. That's all your offspring. You pass away and you're done, okay? So now there was one person in the world that had this mutation, and now your child has it, so there's one again, right? It takes a long time for these beneficial mutations to increase in frequency and become fixed in the population, okay? Doesn't matter how beneficial they are, it takes time, especially in organisms with long generation times like ours, okay? So the probability that this allele would reach fixation, even if it confers an amazing advantage, let's say it gives you a 20% increase in fitness, right? The strength of selection is 20%, okay? Well, the probability that this allele will become fixed ever is only 2 times that, so 40%. Okay, so there's a significant chance that really beneficial mutations exist in populations, but that the strength of selection is just insufficient for them to become fixed for that. They could completely disappear entirely, right? Just due to random chance. We're going to talk about genetic drift and how small population sizes affect allele frequencies, even if those alleles are super beneficial, right? If you have that mutation that makes you resistant to coronavirus, but you don't get your flu shot and you expire due to the flu, right, or pneumonia or something else, then the population's screwed, right? We don't have that mutation any longer. So there are instances where beneficial mutations aren't enough, and they may not ever reach fixation in a population just due to the dynamics of reproductive success and the passing on of alleles. Now, when we think about the trade-offs or the interacting effects of different selection pressures on individuals and their genotypes, it's useful to consider an example like the one presented in your text for these sheep, in which most of the males have this homozygous for the plus allele, the wild type allele, gives them these big burly horns. Super awesome. Okay. That's great for reproductive success. You can fight off other males. The females think you're super amazing looking. You get lots of ladies. Okay. Now the male on the bottom, homozygous for this P allele, they have these ridiculous little vestigial horns that grow kind of funky, don't look great, don't help them fight off other males. Okay. So let's think about the fitness of these two genotypes. Okay. And the heterozygote is also illustrated in the graphs on the right. So relative mating success, that guy on the top with the big horns, the homozygote for the plus allele, the wild type allele, he's got the highest fitness of 1, right? His relative fitness. Remember, there's always going to be a relative fitness of 1. Okay. Now, this guy has the highest mating success, and the individual that's homozygous for the P allele has horrible mating success, right? Only about half what this other guy does. But survival, boy, this guy with the amazing horns has very low survival compared to the heterozygote and the homozygous P individuals, okay? And so when we think about that overall, the relative fitness, okay, it's better to be homozygous for the wild type allele than it is to be homozygous for the P allele, but not by much, okay? Within this population, there's a trade-off between survival and reproductive success, right? If you've got the guy on the bottom has twice the survival and the guy on the top has twice the reproductive success. So it really balances out to where these alleles, both of them, persist in the population. So despite the fact the wild type allele, the plus allele, confers a reproductive success advantage, it doesn't increase in frequency and become fixed in the population. This P allele sticks around because it increases the survival rate of individuals. So this trade-off exists between the P and the plus allele within the population. So when we think about the traits that are under selection, as that previous example illustrates, it's important not to just think about what looks like it's the best, right? Those big horns appear to be the best. Well, in this case, we've got an instance where selection has acted on a population of snails that is variable in their coloration and their pattern, okay? So the snails might be yellow or pink, and they might have stripes or no stripes, okay? Now, because we can observe an instance of disequilibrium in this population on the left, we're going to see something pretty interesting. Look at the population on the left, and you will see that there are individuals with stripes and without stripes, and individuals that are pink and yellow. Okay. Now, there are a couple different combinations there. Okay. There are pink with stripes and pink without stripes. There are yellow with stripes, but there are no yellow without stripes. Okay. That's that disequilibrium, right? That combination doesn't occur. We would expect a roughly equal frequency of all four possibilities: pink with, pink without, yellow with, yellow without. But one of those, yellow without, does not exist, so there's an instance of disequilibrium. Okay. Now, in this case, selection is acting against stripes. Okay. Predators eat all the individuals that have stripes because they're easier to see. Well, if only individuals without stripes survive and they have higher reproductive success, what's going to happen is the population ends up having no stripes. But because the only individuals that have no stripes were pink, the population is also now 100% pink. So selection didn't act on the color, but as a byproduct of selection for no stripes, the color pink became fixed, as did the absence of stripes in this population. So this illustrates the principle of a selective sweep. Okay. And what a selective sweep does is it brings along things that are associated with a trait that is under selection. Okay. So here I've illustrated or put in a figure from your text where we see strands of DNA from 8 different individuals, or 8 different copies of the same chromosome in this population. Okay. These are the only 8 versions of that chromosome that exist in the population. One of them has a beneficial new mutation. The other mutations or positions where there are nucleotide differences are indicated by a blue bar. Okay. So where there's not a dark blue bar or a yellow bar, every chromosome is identical, right? They all have the same things. It's only in these blue or yellow areas where there are differences between them. Okay. Now, what we see here is a beneficial mutation that has arisen. That beneficial mutation is physically connected to this blue bar just to the right of it and this blue bar just to the left of it. These mutations or variants, these alleles at different parts of the chromosome, they're physically connected. So because the chromosome that has this yellow beneficial mutation is going to increase in frequency in the population because it's beneficial, well, because that beneficial mutation happened on a chromosome where there's those other 2 neutral variants exist, as the frequency of that yellow beneficial mutation increases, so too does the neutral variant to the left and to the right of it. And as you can see, over time, the population will become fixed. Every copy of the chromosome in that population will have that beneficial mutation. It will also have those 2 sites on either side of it, left and right, where there's also no variation. The population is also 100% frequency fixed for each of those, to the right and the left. Since this mutation arose, you can see that this beneficial mutation, you can see on the far right that there are additional polymorphic sites that have popped up. Mutations further to the right and further to the left on this chromosome. But they are, of course, all on chromosomes that have that original yellow one and the 2 blue ones on either side that are neutral. Okay. What has happened here is selection has driven that yellow beneficial mutation to fixation, and it has swept with it those 2 on either side of it. Okay. All 3 are now fixed in the population. They have a frequency of 100%. All the alleles, all 3 of these alleles are at a frequency of 100% in the population because of selection acting only on one of those regions. Okay. Now we can see the evidence of this type of selection in cases of artificial selection. Okay. Now the corn that we grow and eat is actually a domesticated version of a plant that traces back to something that still grows in the wild called teosinte. And you can see it illustrated here next to a corn plant. Okay. Now teosinte has all of these multi-branching stems. Notably, corn has a single stem, puts everything into that one stem, and all those corn cobs grow on it. Okay. Now, if we look down below, what we're looking at is you can see those rectangular bars, light blue, green, dark blue, darker green. Okay. That's a portion of a chromosome in the corn and teosinte genomes. Okay. A gene region that has an intron and 2 exons. Okay. Now, what is interesting is if we look at the levels of heterozygosity along this stretch of DNA within the population, that's what these two lines are, the red and the purple plots. All right. Now, what do levels of heterozygosity mean? Let's go back to this previous slide and think about it. Okay. How much heterozygosity is there? I'm moving my mouse around. I don't know if you can see it, but let's look at the very left third of this graph. Okay. Heterozygosity at any one of those sites is going to be high in this population. When I say sites, I mean a place where there is a variant. Okay. When there are multiple copies of a variant, so the one that is labeled neutral variant, right? There are 3 copies of that out of 8 in the population. Okay. So just imagine that means 3 out of 8 copies of that chromosome in the population have the variant. 5 out of 8 do not. Okay. So that's a pretty high frequency. So if you take those frequencies for the variant allele or the non-variant allele, you could calculate what you expect, based on Hardy-Weinberg, the frequency of the heterozygotes to be, right? You could take 3/8 times 5/8 times 2 PQ, to calculate the expected frequency of heterozygotes for that particular portion of the genome. Okay. So you're going to have a lot of heterozygotes. There is high heterozygosity at that locus. Okay. Now let's look at the far right third of this figure. Okay. That same site, how much heterozygosity is there going to be in the population? Where it says site with no variation. If every individual in the population has that neutral variant indicated by the blue bar, are there going to be any heterozygotes for that? No, because there's only one allele anymore. There's no variation in the population. So heterozygosity is 0, crap, nothing. What we see here is how much heterozygosity is there in this region to the left of exon 1, okay, that first light horizontal blue bar, in teosinte. Well, you can see that red line is really high over on the left side of this graph, much higher than it is for corn. What that means is, in the wild ancestor of corn, there was a lot of genetic variation in that region just to the left of exon 1. Okay. Lots of genetic variation, so there are lots of heterozygotes in the population. Heterozygosity is high. But as selection has changed that population to create corn, the variation, the genetic variation in that region has gone down, 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 down to the point where allele frequencies have reached 100%. And when that happens, there's no heterozygosity, right? Because there's only one allele. You can't be a heterozygote if there are no other alleles in the population. So what has happened is Selection has removed genetic variation from this population. So, what does it mean in a population if there's low heterozygosity? So I really want you to think carefully about the influence and importance of heterozygosity on genetic diversity in a population. So I've got a few bullet points with some rhetorical questions here. If we all have the same exact allele, there's no variation in our population, is heterozygosity going to be high or low? Should be able to answer that, okay? If the population is really, really small and there's lots of inbreeding, will heterozygosity be high or low? Now that's a more difficult question, right? But I want you to think about what heterozygosity means. It means having two different alleles for the same gene, okay? Now, think about what inbreeding means and why, for example, humans don't inbreed, or we frown upon inbreeding our dogs. Why? What's the consequence of inbreeding? The consequence of inbreeding is the increased chance that you get a crappy allele from one parent and the same crappy allele from the other parent, because they're so closely related, they both have that same crappy allele, and you get two of the same thing, okay? You are going to be homozygous for that locus. And so when populations are small, that leads to, out of necessity, higher levels of inbreeding. When you have higher levels of inbreeding, you're more likely to get two copies of the same allele because you have such close relatives that are passing that same allele down to you, okay? So again, in small inbred populations, heterozygosity is going to be low. And heterozygosity in general is not good. I'm sorry, when heterozygosity is low, it's not good, okay? Heterozygosity being high means there's lots of genetic variation, okay? And it should make sense to you now that for the fitness of a population, it's good to have lots of genetic variation, okay? So if we want a population to be genetically diverse and have lots of genetic variation, that could allow it to adapt to many environmental changes. Are we going to favor high levels of heterozygosity or low? Okay. So read this section in the text. I want you to think critically about heterozygosity. Now, we're going to talk about some specific instances where fitness is different for the heterozygote. Fitness of the heterozygote can either be the highest or the lowest for any genotype. In overdominance, what we have is a scenario where heterozygotes have the highest fitness, higher than either that should say have higher fitness than either homozygote, not heterozygote, okay? So overdominance is when heterozygotes have the highest fitness. And the example here is one you've almost certainly seen to this point. This is sickle cell anemia, right? Where the allele that causes it is most common in Central Africa, Madagascar, and Saudi Arabia, okay? And it's also not shown on this map, pretty high in India. That allele, okay, when you are homozygous for it, causes all your red blood cells to have this sickle shape. They don't do great at transferring, carrying oxygen, and it's a very bad genotype to have. It causes serious health issues, okay? But this S allele, as you can see, is quite common across much of Africa. Why is that? Because compare that to the map of where malaria occurs and where it's most common. The presence of the S allele, those sickle-shaped blood cells, those actually are resistant to malarial infection, okay? So with this resistance to malarial infection, the frequency of the S allele has stayed pretty high throughout Africa because of this principle of overdominance. Now, the combination of the frequency of an allele and its fitness can determine the fate. Now remember, I've told you that it could be the best allele in the world, but if its frequency is really low, it doesn't mean it's going to reach fixation. So we've got 3 different scenarios here where the frequency of the A2 allele, it starts out at either 10%, let's say 40%, or 70% in this top right graph, okay? And over time, in an instance of positive selection for A2, where A2 is the best, you can see the frequency of the A2 allele increases and reaches fixation pretty quickly. In instances of overdominance, okay, what I just talked to you about, you can see that the heterozygote, the W12, W being relative fitness, the relative fitness of the 1-2 heterozygote is the highest, okay? In overdominance, the heterozygote has the highest fitness, and look at these 3 different scenarios for the frequency of A2. A2 could start out damn near 0 or at 1, and either way, it's going to converge on this intermediate frequency of just below 0.4, okay? This is because the A2 allele can't reach a frequency of 1. If it did, then all the individuals would be homozygous for the A2 allele and could not, they would have lower fitness than the heterozygote. All right. So the third instance, underdominance, is a scenario where the heterozygote has the lowest fitness. It's worse to be a heterozygote. It's better to be either homozygous for A2 or homozygous for A1, either one. One is better than the other, but the population is going to go to one of those. All right. And depending on where the A2 allele frequency begins, affects where the population will end up. You can see if that A2 frequency starts low, let's say around 30%, A2 disappears. Okay? What happens when A2 disappears? It means A1 is at a frequency of 1. It's fixed. The whole population is going to be homozygous for A1. Okay? Now, if the A2 allele frequency starts at 0.4 or 0.8, of course, you can see that the population rapidly increases to a high frequency of A2, becoming fixed for that allele. Now, one of the things I want to make clear is that fitness might vary within a species. And this isn't all that different from the example I showed you with the alleles for human skin color being, for light skin color being common in Europe and not common in Africa. It's because the environments are very different in those areas. This example from your text illustrates mosquitoes that were sampled along a road. So if you follow all these little tiny black dots on the map of Cameroon, you will see that this is a road. Okay, so some biologist drove this road and caught mosquitoes all along the road, the length of Cameroon. You can see that if you drive that road north to south in Cameroon with the elevation superimposed on this map, where it's darker, it's higher elevation, you can see that the road goes up in elevation and then back down as you go north to south. All right. What's interesting is there's a region of the chromosome that is illustrated on the far left where there's an inversion, a flip. Okay. That chunk of the chromosome has been cut out, flipped, and pasted back in. Now, that inversion can occur in an individual. So the little blue, white, and black color codes here indicate whether you are homozygous for the inversion, that's black, heterozygous, where it's S and I, okay, that's blue, or homozygous for not having the inversion, white, homozygous for S. You can see that the white genotype is most common in the north. The homozygous genotype for the inversion is most common in the south. and then the heterozygote is most common in the mountainous areas in between. So what this suggests is that there is a gradient, a selection gradient, where one of these genotypes is favored in the north, the homozygous for the non-inversion, and then homozygous for the inversion in the south. Okay. The next thing I want to touch on is selection that is dependent upon the frequency of the genotype. Okay. If that genotype is most common in the population, then frequency dependent selection would give it the highest fitness. If the genotype is less common in the population, then under negative frequency dependent selection, the fitness, the rarer it is, the better. Okay. So positive frequency dependent selection and negative frequency dependent selection are situations where the fitness of a genotype is entirely dependent on how abundant it is in the population. Okay. So positive frequency dependent selection, the example here is a coral snake, where when coral snakes exist in a population, they have this really bright coloration. That bright coloration is meant to warn predators that it would be disadvantageous for them to interact with that venomous snake that could hurt them. Okay. And so the more common that signal is in the environment, the more wary potential predators are going to be about approaching one of these snakes. Okay. They've learned it. They know it. They avoid it. So the more common it is, the better it is for those snakes to have that color. If, on the other hand, that snake is the only one to have ever had that coloration, no predators recognize it, and it is not going to be beneficial, right? If you stick out like a sore thumb and nobody has any idea what those bright colors mean, it would be beneficial. It would not be beneficial. Okay. Negative frequency dependent selection is the opposite. Okay. Where the less common the phenotypic trait is, the higher the fitness is. So the rarer, the better, right? And there was a really interesting study that came out a few years ago about the increased prevalence of beards. And there's a picture of George Clooney growing a beard that was associated with the article. And it was interesting to note that in human populations, as the frequency of beards declines, then beards become cooler and potentially more attractive. And so then more and more men start growing beards because it's beneficial. But then when everybody has them, okay, the more common it is, then it's not as beneficial. Okay. So these are two different examples of selection regimes in which the fitness of a phenotypic trait is entirely determined by how abundant that trait is. Okay. And there can be a direct relationship between abundance and fitness, right? The more abundant it is, the better it is. Or there can be a negative or an inverse relationship between the frequency and the fitness, okay? Whereby the more abundant it is, the lower the fitness, okay? And I'll go through two examples here, presumably, if I get this to advance. So the instance of negative frequency dependent selection that your text uses is a species of plant that has two different colored flowers, yellow or purple. And the x-axis on these two graphs is the frequency of the yellow plant, all right? Now, if we look at the top graph, that is the relative male reproductive success. So how many of its pollen are removed by insects that come to that plant, okay? Well, you can see that as the frequency of the yellow plants increases, the proportion of their pollen that is removed is decreased. Okay. So that's the first step in reproduction. Those male plants have to get their pollen taken from their flowers to a female. So with this negative relationship, what we are seeing is these bees that pollinate these plants, when they go to these plants, realize that they're going there hoping that they will get some kind of reward, some kind of nectar or something, let's say. Well, when they go to these plants and they are not rewarded, if most of the plants that they are going to are yellow, then they are going to remember, I'm not going to any more yellow plants. I'm not carrying away that pollen. So the more plants that are yellow, the quicker the bees learn not to go to them, and the fitness of yellow plants declines. Now, the same thing can be seen on the bottom graph, the relative female reproductive success. As there are more and more yellow plants, okay, there are fewer and fewer females having reproductive success. Okay. So again, this is an instance of negative frequency-dependent selection, where fitness is inversely related to frequency. All right. So you can see that in that top graph. Frequency goes up, fitness goes down. Okay. Now, a positive frequency-dependent selection, the example I used before was a coral snake. This is an example drawing on the bright coloration of poison frogs, where a bird predator might need to learn not to consume these brightly colored frogs. So they try a couple of them and decide, you know what, I'm never tasting one of those again. But they have to learn by tasting one or two or three or four. Okay. It takes a while for them to learn. Okay. So they grab them, spit them out. And after a couple of tries, they're like, forget it. I'm never trying a yellow and black frog again. Okay. So because of that high frequency of this phenotype in the population, you know, the vast majority of individuals never even experience an attack by a bird. So the population enjoys this higher fitness. However, what if you are the really rare, unique looking individual in the population? You've got some mutation, your phenotype is quite different than everyone else. Well, under those circumstances, because you are so unique, none of the birds in the population have tried anything that looks like you. And every single one of them will think, I'll give that a shot. I've never tried a green frog with black spots. Okay. So because of this low frequency, this phenotype enjoys terrible fitness. It has very low survival rate because of its low frequency. And so under this scenario, the fitness of a phenotype is perfectly related, it's positively related to its frequency. The higher the frequency, the higher the fitness. Okay. An example from your text is illustrated in 524, and this is a really remarkable scenario that has been studied extensively by evolutionary biologists. And this is one in which two species of butterfly, two different species, live in the same places throughout much of South America. And everywhere you go, the butterflies, the two different species, look just like one another. And what's remarkable is they look pretty different everywhere you go. So each of these places, it's illustrated what those two species look like. So Heliconius melpomene is the one on top, and Heliconius erato is the one on the bottom. So those are the two different species, top and bottom. But everywhere you go, both those species look the same. But every different place, they look different. Okay? The reason the two species look like one another, and we call that mimicry, the reason they look like one another is selection. It's beneficial to them if more individuals look like that. Think back to that example of the bird with the black and yellow frogs. Imagine, you know, I said that bird had to try 4 frogs, right? That's tough on a species if every bird has to try 4 frogs. What if they have to try 4 butterflies? Well, now these two species share that burden. Okay, they might have to try 2 melpomene and 2 erato. It's cut the training burden in half. And so each species does even better because of this mimicry. So that the frequency of these phenotypes is very high in the population because there's more than one species that has the same phenotype. Okay. Towards the end of the chapter, the text hits again on the fitness of different genotypes, positive selection versus overdominance and underdominance, something I've already talked about right down the middle here. Populations in general, under any of these circumstances, are going to evolve towards a higher mean fitness for the population. And what that means is, depending on how selection acts on the different genotypes and the different alleles, right? Whether that beneficial allele is dominant or recessive, you can see that the frequency of those alleles and the fitness of the population changes differently depending on whether it's dominant or recessive. And in instances of overdominance and underdominance, these two graphs make some really interesting illustrations that weren't clear in the previous slide that I showed you, or they didn't quite show this. This is showing the frequency of a particular allele P in instances of overdominance and underdominance. And so what's going to happen is whatever this allele P is, in this case, let's say it's W2, the frequency of P in a case of overdominance, if the allele P starts out anywhere from 0 to 0.5, you can see that allele frequency is going to increase until it gets to about 0.6. If that allele frequency starts anywhere between 0.6 and 1, that allele frequency is going to shift towards 0.6. Given the specific example illustrated in the center graph of overdominance, that the allele frequency P is going to find a perfect balance somewhere around 0.6, where P and Q, the two alleles, persist in the population, just like that sheep example I described to you of overdominance. We are going to have an intermediate value of P and one of Q as well. But the opposite happens with underdominance. When the heterozygote has the lowest fitness, the frequency of P is going to shift either to the left or to the right. It's either going to reach 0 or 1 relatively quickly. So that same principle is illustrated here in different examples of positive selection or overdominance and underdominance. And I encourage you to think about the frequency of these alleles, A2, with respect to where they started. That's the arc of the blue line. The frequency of A2 could start anywhere on that blue line. But under these three different circumstances of selection, that frequency of A2 is going to shift to one of those solid blue dots, okay? And it's going to shift due to these selection pressures exerted by these instances of overdominance or underdominance. Okay, and now the last point I want to make in chapter 5 is that mutation, evolution, and selection doesn't always result in increasingly higher and higher and higher and higher fitness for a population. And I just want to make that clear here because I don't want you to leave this class thinking that evolution is this perfect mechanism where everything just gets better and better and better and more fit, okay? This is a really cool example where a population of plants produces fruit, right? So this ancestral population, these really low shrubby plants produce loads of fruit. They have high fecundity. In B, we have a mutation that arises in the population that causes some individuals to grow tall, woody stems, okay? But as a consequence, they don't produce as many fruit. They have lower fecundity, lower reproductive output. But what they are doing also is they are shading out those short plants in between them. So they end up having higher survival, and they could, over time, the population evolves to just be tall, woody trees that produce fewer offspring. Okay, so the overall fitness of these populations, this population of fruit trees, is probably lower than it was originally, okay? But because of this mutation that arose, the population evolved, and there was really no other way for this to go down other than the population to decrease in fitness due to the mutations that arose, okay? So, and it's the calculating that absolute fitness for the short trees versus the tall trees. What happened was, as the circumstances changed, that probability of survival changed for the short trees because the large trees shaded them out. Okay, so how do bad mutations disappear from a population? Okay, well, at this point, you should realize that the way that bad mutations, deleterious mutations, are removed from a population is through selection, right? Through low survival, low reproductive success. But I've told you that populations, they're not all perfect right now. Why not? Well, it's because that additional mutations are constantly reintroduced through this mu down here. Mu represents the mutation rate, okay? Mutations are happening all the time. Selection is happening all the time. Selection gets rid of bad stuff in general. It will remove deleterious alleles from the population. Allele frequencies for the better allele will approach 1, as we've seen in many instances in this lecture. And so as allele frequencies for the good alleles reach 1, necessarily the bad or deleterious mutations are gone. They're 0. But populations aren't perfect because there are constantly new mutations being introduced into the population. And so the frequency of a particular allele in the population, its persistence and frequency in a population, is in part determined by the mutation rate and the strength of selection against that mutation, okay? Now, if the mutation rate's really high and the strength of selection against it isn't that low, then you're going to have a pretty high number here, right? If that numerator is much higher than the denominator, you're going to have a pretty high frequency of this allele. Conversely, if the mutation rate's really low and selection is really high, you're going to have a small numerator, a large denominator, and that frequency of that allele is going to be really low, okay? So the text goes through an example with the numbers that are here, but as you can see, with even low selection, 1% selection acting against a particular mutation, and this is an average mutation rate, you can end up with a pretty low frequency of this allele persisting throughout time. So even though selection might act against a mutation, if the mutation rate's high enough, it'll stick around in the population. Okay, that's all I've got for chapter 5. I'm going to post chapter 6 here shortly. And if you have any questions, don't hesitate to email.